Hello everyone, welcome back to the sessions bioinformatics discussion sessions. Uh, Today I am going to host the session. So in this session, Ranjan Behera, a same student of BJP College, is going to represent on molecular docking. Okay, start.
So, good evening, everyone. And molecular docking. Anyone has any idea about molecular docking? Like anything? Anyone? No one?
जो
So,
मैंने थर्ड डे सेशन रे जॉइन करथिला सेठी भी तो molecular docking सर कुआ थिला. So anyone want to answer?
किच्छी तो गोटे किच्छी बेसिक्स भी चलेगा. Anyone? कुछ बेसिक्स भी चलेगा.
चार्ट रे जति परिबो कहि परिबो.
हम्म. मोर स्क्रीन देखा जाउछ तो.
स्क्रीन देखा ने रिप्रेजेंट करना था.
करेनी.
कंप्यूटर वाला ड्रॉप डिजाइनिंग. ओके समीर.
रॉटिक के रो. हेलो.
हम्म
Right. Okay. So, molecular docking. So,
टाइम ऑफ फिट टुगेदर मॉलिक्यूलर एंजाइम.
hello everyone. My name is Ranjan Behera, as introduced. मैं आपसे मॉलिक्यूलर डॉकिंग के बारे में कुछ शेयर करना चाहता हूँ कि मॉलिक्यूलर डॉकिंग मतलब सिंपल वर्ड्स में बोला जाए तो इट्स लाइक फाइंडिंग अ कंपैटिबल मॉलिक्यूल. जैसे कि मान लो कोई भी मॉलिक्यूल को एक्टिवेट या फिर उसको कैसे बोलते हैं कैटलाइजिंग करने के लिए एक स्मॉल मॉलिक्यूल की ज़रूरत होती है जो होती है लाइगेंड, और एक प्रोटीन जो अपने आप को और ज़्यादा एनहांस करके परफॉर्म करवाने के लिए लाइगेंड के साथ इंटरैक्ट करके जो मॉलिक्यूल बनाता है, तो वो होल प्रोसेस को बोलते हैं डॉकिंग. सो बेसिक आइडिया ये था. तो पहले कंटेंट्स में हम लोग देखेंगे व्हाट इज मॉलिक्यूलर डॉकिंग, देन वी विल फॉलो विथ व्हाई वी नीड मॉडलिंग, लाइक व्हाई वी नीड सो, and what is molecular docking and classification about it, then categories of docking, then what are docking and scoring.
समीर
um,
प्रेजेंट होने
हाँ,
ब्लैंक আসছে.
blank আসছে.
हम्म
Erotic.
भाई किसी आसुनी
Good second.
आराम से.
ହେଲା
हाँ
ଆସିଲା
येटा करतो माने तो पीपीटी टा देखा या जोटा तू शो करिया ପାଇଁ ଚାହୁଁ.
ଆସିଲା.
बस बस परफेक्ट.
ए टेबल पर देख
निहारिका.
Okay, so here are the contents, like what is molecular docking, then we'll go with why we need modeling and molecular docking classification, then we'll go for categories of docking, and what are docking and docking scoring, then we'll go with available docking programs, then with components of docking software, like what all softwares are present, and like what are the basic steps to like carry out a process, then docking flowchart using Autodock, best known, then application of molecular docking in modern drug discovery, Then evidences of success, like how much success rate according to molecular docking is available in the uh, what to say in the real world. And then conclusion. Okay, so what is molecular docking? So molecular docking typically involves like predicting binding conformation and affinity of a small molecular um, what to say a small molecule to a larger one. Like the small molecule is the ligand and the larger one is the protein. So docking is a computational determination of binding affinity between molecules. Given a protein and a ligand, find out the binding free energy of the complex formed by docking them. Why we need modeling? Like ये सब जो हो रहा है हम लोग क्यों कर रहे हैं? Experimental determination of the structure is still a time consuming and expensive process. Like अभी मान लो हमको किसी का स्ट्रक्चर अभी पता करना है, तो अभी अगर हम लोग जाएंगे वेट लैब, we'll perform various tests, we'll conduct various tests and further, but at real time we won't be getting the structure that we really need at that time. But through computational methods it would be easy and it would be like real time saving for getting a structure. As we know, the sequences are already known and which are more than the known sequences, like known structures. We don't know the structure, we only know the sequences. Then we perform various steps, various processes to get to know the structures that we are what we are opting for. And as it mentioned that structure information is essential in understanding function. So molecular docking classification. Molecular docking or computer aided drug designing can be broadly classified into receptor based methods. Make use of structure of the target protein. Like we have the structure of the protein and we'll use that to find the ligand which is essential for the activation of the protein, or ligand based methods based on known inhibitors. Like how to say a protein is consisting of inhibitors, so we'll use that to perform to carry out various processes and to get a ligand. Receptor based methods, so uses 3D structure of the target receptor to search for the potential candidate compounds that can modulate the target function. These involve molecular docking of each compound in the chemical database into the binding site of the target and pretending the electrostatic fit between them. So molecular docking means compatibility as mentioned. So as each of the compound needs to be like compatible enough to reduce the electrostatic what to say energy between them and to become more more tightly packed so as to fit inside what to say drug, vaccine, anything you can say. These compounds are ranked using an appropriate scoring function, such as the score correlate with the binding affinity. And receptor based methods have been successfully applied in many targets. So this is the like applied and tested method. So this is ligand based strategy. In this, the absence of structural info of the target, ligand based methods make use of info provided by known inhibitors for target receptor. Structures similar to the known inhibitors are identified from chemical databases by a variety of methods. You can use several methods to like get to know about the inhibitors. Some if some of the methods widely used are similarity and substructure searching, pharmacophore or 3D shape matching. Numerous successful applications of ligand-based methods have been reported. So, categories of docking: protein-protein docking. Both molecules are rigid. Interactions produce no change in conformation. Protein ligand docking. Ligand is flexible, but the receptor is rigid. Interaction produces conformational changes in liquid. So there is one more, like ligand also is there, but that's like, what to say, um, not that categorically important, but these two are. So I'll just discuss about them. So protein docking. So computational modeling of quaternary structure of complexes formed by two or more interacting biological molecules. These are used, like protein docking are like using of bigger molecules rather than like secondary or tertiary. They use quaternary structures. Protein complexes are the most commonly attempted targets of such modeling, followed by protein nucleic acid complexes. So this is the image that you can say, where two proteins after docking form a complex or a bigger protein called complex AB. Okay, so protein ligand docking. Here the target is like the protein, and the ligand is a smaller molecule that fits to the protein, and they form a what this docked molecule. So protein ligand docking is to predict the position. Protein ligand docking is to predict the position and orientation of a ligand, a small molecule, when it is bound to a protein receptor or enzyme. This was very clear, I must say, and you might have got it. So, what are docking and scoring? So, molecular docking means uh, usage of computational methodologies into the binding site of a receptor or in the manners of appropriate for optimal interactions with the receptor. But molecular scoring is used to evaluate the ligand receptor interactions in a way that may discriminate the experimentally observed mode from others and estimate the binding affinity. So, in short, molecular docking is the computational method, while molecular scoring uses mathematical methodologies to like carry out processes. So this is the thing, like this is the ligand, and this is the receptor. After docking, they form these complexes, and scoring these complexes, we get X-ray structures and various other like you want to find any kind of what to say, weight or anything you can use through molecular scoring. So here are some available docking programs like Schrodinger, Dassler's Pro, Gold Dock, Moedock, MOE Dock, Flex, Autodock, the most commonly used, Fred, Hammerhead, Argus Lab. So components of docking software. So typically protein ligand docking software consists of two main components which work together, like search algorithm generates a large number of processes of a molecule in the binding site, and scoring function calculates the score or binding affinity of uh, of a particular pose, the binding affinity or a score representing the strength of the binding, like how well a molecule is bind with the ligand and form a complex one. Okay. So this is the docking flow chart, as you can see on the left hand side. First, we need to identify the protein of interest that we need. Then ligands are identified. Then virtual screening by Pyrex. You can use it like you can use various other tools also, but Pyrex is like you can rely upon. Then drug likeness properties analysis, like which is both of them are compatible or not. Then docking, Autodoc Vina. Then structure visualization through PyMol. Like you can use Rasmol, but I would suggest you go with PyMol. That's more efficient, I would say. So Ligand preparation for docking using Autodock. Okay, so you first need to assign charges to the ligand. Then you need to define the rotatable bonds. I'm sorry. Rename aromatic carbons, and then you have to save it like dot pdbqt ligand file. In that way, you have to save. Then preparation of protein using Autodock. You have to add essential hydrogens like polar ones. Then you have to load charges. Kolfman, some I don't remember the charge. Remove water molecules, and then you have to store this also in .pdbqt file. Okay, so before going to this, I would like to show you the process. Hmm. Okay, so here I have taken the protein. This one I found it in the PDB. You can take any protein of interest, and then you have to just enter the ID. Then you'll get this. You can analyze everything over here. Like you can find the chains, then the sequence length, organism, etc. Details, images of the chain A. You can find it. Okay, so after this, you have to download it in FASTA in PDB format, as it I have downloaded it earlier, but still. Okay, so after that, you have to open Autodock. Like I'll provide you the link where you can just go and download this. So after downloading, you have to just drag and drop here. So as you can see the protein structure. So this is the protein structure. First, we have to just remove any water molecules because this would cause any hindrance in the interaction. Then we have to add hydrogen, polar only. As you can see, it has just added the hydrogen molecules here. Then you have to add charges. So this is the charge that has been added, the Coleman charge, as I was talking about earlier. So you have to add. So this is the structure that you got. So we have to like save this to perform the docking. Okay, so we have to save it where we have downloaded the format. Okay, I have downloaded here. As I have it over here, I'll just skip to the next part. Okay, so now as we have the protein uh, protein structure, now we have to find the ligand structure. So we'll go with the okay. Okay, so this is the ligand structure that we need to find. Okay, wait. Okay, so in after so to find the ligand, you have to come to PubChem. There you have to like type um, any inhibitor you want or stuff. Then you have to just go through. Then you have to come here, come down, like download coordinates. Then you have to search for SDF. Then you have to save because you need to carry out in Pymol. Okay, so Pymol. Okay, so after Pymol, you need to like you have to open. So this is the ligand structure that you're gonna get. Okay, so after this, export molecule enabled. You just have to save it in PDB format where you have your other folders. Like I have it over here. So I have it, so I won't save it again. So I'll just show you how it's like. Okay, so let's open it again. Okay, I need to cut this out. So now you have both the files in PDB format. So now you have to take the ligand. Hello, hello, hello. Oh, great. हेलो सुबिला मतलब ये देखा जाଉଛି
स्क्रीन टा फ्रीज ହେଇଯାଇଛି ଷ୍ଟପ୍ ଇଏ କରିକି ଆଉ ଥରେ ପ୍ରେଜେଣ୍ଟ କର
ହେଲା ଠିକ ଅଛି ନେଟୱାର୍କ ଇସ୍ୟୁ ଅଛି
ଆଚ୍ଛା ଏ ଭିତରେ ମୁଁ ଗୋଟେ କ୍ୱେଶ୍ଚିନ୍ ପଚାରୁଛି ଜୁନିୟର ଯେଉଁମାନେ
ଅଛି
ପୂର୍ବରୁ ଇଏ ଅଛ ସେ ତୋ ପାଇଁ ନୁହେଁ ତୁ ସବମିଟ୍ କର ମୁଁ ଏପଟେ ଏମାନଙ୍କୁ ପଚାରୁଛି
ହୁଁ
ଆଚ୍ଛା
ହୁଁ
ଆମେ ଏଠି ଫାଷ୍ଟା ଫର୍ମାଟ୍ରେ ନ ଆଣିକି ପି ଡି ଏଫ୍ ଫର୍ମାଟ୍ରେ କାହିଁ ଆଣିଲେ ଡଟ୍ ଫାଷ୍ଟା କରିକି ତ ଆମେ ଯେତେ ବି ଏପର୍ଯ୍ୟନ୍ତ କାମ କରିଛନ୍ତି ଏ ଭିତରେ ତ ଆମେ ଫାଷ୍ଟା ଫର୍ମାଟ୍ରେ ଧରିକି ଆସିଛନ୍ତି ଡଟ୍ ପି ଡି ଏଫ୍ ଫର୍ମାଟ୍ରେ କ'ଣ ପାଇଁ ନେଇକି ଆସିଲେ ଏନିୱାନ୍ କ୍ୟାନ୍ ଆନ୍ସର୍ ଆମେ ଆଗରୁ ଯେତେ ବି କରିଛନ୍ତି ନା ସବୁ ଫାଷ୍ଟା ଫର୍ମାଟ୍ରେ ଧରିକି ଆସିଛନ୍ତି ନା ଏଇଠି କିନ୍ତୁ ଆମେ ଫାଷ୍ଟା ଫର୍ମାଟ୍ ନ ଆଣିକି ପି ଡି ଏଫ୍ ଫର୍ମାଟ୍ ଧରିକି ଆସିଛନ୍ତି କ'ଣ ପାଇଁ ନୁହଁ ଆଚ୍ଛା ଫାଷ୍ଟା
ଟିକେ
ଫର୍ମାଟ୍ରେ ଆମର କ'ଣ ଥାଏ କୁହ ତ ଫାଷ୍ଟା ଫର୍ମାଟ୍ରେ ମୁଁ କହିଥିଲି ଯେଉଁ ଫାଷ୍ଟା ଫର୍ମାଟ୍ରେ କ'ଣ ଥାଏ
ଟିକେ ମୁଣ୍ଡ ଖର୍ଚ୍ଚ କର
ଫାଷ୍ଟା ଫର୍ମାଟ୍ରେ କ'ଣ ଥାଏ କେହି ବି ଆନ୍ସର୍ କରିବନି हर्षिता बरार हाँ we need structure maybe that's why. Okay, absolutely मतलब କ'ଣ ନା ଦେଖ ଆମେ ଯେତେବେଳେ ଫାର୍ଷ୍ଟ ଫର୍ମାଟ୍ରେ ଧରିକି ଆସୁ ଫାର୍ଷ୍ଟ ଫର୍ମାଟ୍ରେ ଆମେ କ'ଣ ନେଇକି ଆସୁ ଓନ୍ଲି ସିକ୍ୱେନ୍ସ ଆଉ କଣ୍ଟିକ୍ ସହିତ ସିକ୍ୱେନ୍ସକୁ ଆମେ ନେଇକି ଆସୁ ଏଇଟା ମୁଁ କହିଥିଲି ତମେ କଣ୍ଟିକ୍ ନା ପରେ ଯାଇକି ଚେଞ୍ଜ କରିବ ସେ କିଛି ପ୍ରୋବ୍ଲେମ୍ ନାହିଁ ଠିକ୍ ଅଛି ବଟ୍ ଏଠି କ'ଣ କରୁଛନ୍ତି ନା ଆମେ ଷ୍ଟ୍ରକ୍ଚର ନେଇକି ଆସୁଛନ୍ତି ତ ଫାର୍ଷ୍ଟ ଫର୍ମାଟ୍ରେ ଆଣିଲେ ତ କେବଳ ସିକ୍ୱେନ୍ସ ହିଁ ତ ଆସିବ ଆମକୁ ଷ୍ଟ୍ରକ୍ଚର ଥୋଡ଼ି ନା ସେ ଦେବ ପିଡିବିକୁ ଯାଇକି ଆମେ ଯେତେବେଳେ ସେଟାକୁ ଡାଉନଲୋଡ୍ କରୁଛନ୍ତି ସେଥିପାଇଁ ଆମେ ଷ୍ଟ୍ରକ୍ଚର ଫର୍ମାଟ୍ରେ ତାକୁ ଡାଉନଲୋଡ୍ କରୁଛନ୍ତି ତ ଆମ ଷ୍ଟ୍ରକ୍ଚର ଫର୍ମାଟ୍ଟା ହେଲା ଡଟ୍ ପିଡିବି ଓକେ ସିମ୍ପଲ୍ କ୍ୱେଶ୍ଚିନ୍ ଥିଲା ଭେରି ଗୁଡ୍ ହର୍ଷିତା ହେଲା ତାର ସମୀର
ହେଲା ଆସୁନି ତ ସ୍କ୍ରିନ୍ ସେୟାର ହଉନି ଆଉ
ସେୟାର ହଉନି ଆଉଥରେ ନହେଲେ ବାହାରି ଯାଇକି ଆଉଥରେ ଜଏନ୍ କରିଥିଲେ ହଁ ସେୟାର
ହେଲା ହେଲା
ହଁ ହଁ ହଁ
ତ ମୁଁ କୋଉଠି ଥିଲି ଆଚ୍ଛା ହଁ ହଁ ତ ଇଣ୍ଟରେଷ୍ଟିଂ କ୍ୱେଶ୍ଚନ୍ ଥିଲା କି मतलब କାହିଁକି ଆମେ ପିଡିବି ଫର୍ମାଟ୍ରେ ନଉ ଆନ୍ସର ସେ କରିଥିବ ବଟ୍ ମୁଁ କହିବାକୁ ଚାହିଁବି କି ଦେଖ ଫାଷ୍ଟ ଫର୍ମାଟ୍ରେ ସିକ୍ୱେନ୍ସ ନିଆ ହେଉଛି ସୋ ଓନ୍ଲି ଦ ଲେଟରିଂସ୍ ଆର୍ देयर कॉमन सेंस ଟିକେ ଲଗେଇଥାନ୍ତ ଲେଟର ଅଛି ସେଠି ତମେ 3D ଷ୍ଟ୍ରକ୍ଚର ଯେତେବେଳେ କାଢୁଛ ତ ପିଡିବି ଫର୍ମାଟ୍ ନେଇକି ହିଁ କାଢିବ ନା ଫାଷ୍ଟ ଫର୍ମାଟ୍ରେ ସେଟା ପେଷ୍ଟ କଲେ କ'ଣ ହିଁ ଆସିବ ବୁଝିପାରିଲ So this was the common answer that you might have just put forth. But anyways, we'll go. କେତେ ଯାଏଁ କରିଥିଲି କି ଆଚ୍ଛା ସୋ ନାଇଁ ଗୋଟେ କରି ସାରିଥିଲି ରାଇଟ୍
ଏଟାକୁ ସେଭ୍ କରିସାରିଥିଲୁ ମାନେ ଲାଇଗେଣ୍ଡଟାକୁ ସେଭ୍
ସେଭ୍
କରିସାରିଥିଲୁ ଏବେ ସାଇଡ୍ ଇଣ୍ଟରାକ୍ସନ୍ ଷ୍ଟଡି କରିବା
ତ ଏଟା ହଁ then after like converting it to the PDB format the ligand is ready to be auto docked so just yeah. So we'll just drag the ligand PDB format over here. So this is the ligand in PDB format. And we have the protein too in PDB format. So first we need to convert this thing into PDB QT format. So we'll just from here, okay. So after like grid macromolecule, choose ligand, select molecule. Okay, then we have to save. The same procedure goes with this also. Okay, so we'll get a PDB QT format. Then after this, we'll again open. Wait, need your time. Okay, so we'll open another table. So after reducing the ligand and the protein one, we'll get like ligand PDBQT and protein PDBQT files. So we need to drag both of them. Now, as you can see, so this is the interaction, like this is not the full interaction, but the full interaction might take some time. And so this is how the ligand needs to be attached. This is the ligand, this is the protein molecule, so it needs to be attached in this format. But due to some technical issue, I need to just go through it once again. So yeah, this is how Autodoc works. So for Autodoc, you need to like download two more files, two more softwares, like two more softwares like what to say, one is Pymol and the other one is and the other one is MIGL tool. These two you have to download, then only you can perform Autodoc. Okay, so let's get back to the PPT. Okay, so application of okay. So application of molecular docking in modern drug discovery. As we all know that drug discoveries are taking place in a rapid format. So determining the lowest free energy structures for the receptor ligand process complex, search database and rank hits for lead generation, um, calculate the differential binding of a ligand to two different macromolecular receptor, study the geometry of a particular complex, propose modification of lead molecules to optimize potency or other properties, de novo design for lead generation, library design, of course, screening for the side effects that can be caused by interactions with other proteins like proteases, cytochrome P450, and others can be done. It is also possible to check the specificity of the potential drug against homologous proteins through docking. Okay. The docking is also a widely used tool in predicting protein-protein structures. As mentioned earlier, knowledge of the molecular association aids in understanding a variety of pathways taking place in the living and in revealing of the possible pharmacological targets. Docking based virtual high throughput screening. This is very important. Less expensive than high throughput screening, obviously. Faster than conventional screening. Scanning a large number of potential drugs like molecules in very less time. HTS means high throughput screening itself is a trial and error approach, but can be better complemented by virtual screening. So these goes hands in hand. So finally, we draw the conclusion that molecular docking gives the promising effect on identification and optimization in modern drug discovery. The combination of the chemical information of natural products with docking based virtual screening will play, I'm sorry, like an important role in drug discovery in the post genomic era, as more and more new potential targets are emerging from the functional genomic studies. As we know that various kinds of diseases are approaching humans like anything. So through this we can save time. Through this we can just perform heavy tasks in shorter period of time, and we can get results immediately. So docking based virtual screening lead to much higher hit rate than traditional screening methods, as mentioned. Docking methods provide an opportunity for the designing of active compounds, as we can see that which one is less active or which one is more active, and by attaching it with a ligand, how much the activity rate increases, that can be determined. However, it has been emphasized that docking based virtual screening is not the replacement of the actual experimental screening. Obviously, like the process that we can handle in wet labs are much more appropriate than the virtual screening, but due to less consumption of time and like uh, what to say, to get more focused on threats or stuff. So this is more reliable than the wet lab performance, as it is uh, much time taking. As a matter of fact, these two methods are highly complementary, obviously, because after screening we have to perform that stuff in uh, what to say, wet labs, so as to get to know that the process or the method that we carried out in in silico method is perpetual or not. So thank you everyone for listening to my PPT and my voice and my speech and everything. So any questions you have, you can kindly share it.
Hello. Hello.
हाँ रे कह.
So post genomic era या विषय କିଛି କହିପାରିବୁ?
पोस्ट जेनोमिक एरा इन द सेंस, देख पोस्ट जेनोमिक, पोस्ट माने कौन? हाँ तो पोस्ट जेनोमिक एरा माने कौन है?
पोस्ट जेनोमिक एरा मीन्स एबे जेनोमिक उपरे आउ किछि सेते खोजिबा दरकार नै कम ना सेटा डेवलप होई सारछे नेक्स्ट जनरेशन सीक्वेंसिंग वगैरह थ्रू रे तो हम पाखा एबे जेनोमिक सीक्वेंस प्रोवाइडेड अछि राइट तो एबे
हाँ
आमे हमको माने कोन किछि भी गोटे होमोलॉजी मॉडलिंग तो धरिले कोन से गोटे प्रोटीन को करिया पाए तो आमे आउ जेनोमिक सीक्वेंस मिलिबोनी बोली आउ आमे सही ये रे रहबा राइट हमको एबे कोन हम ए एरिया रे अछले जोडे कि कोन सीक्वेंस गुडा हमरा प्रोवाइडेड अछि ओके
देख तो पाखरे जदि भी मतलब कौन देख कॉम्प्लीमेंटरी मु सेत पे कहली देख सीक्वेंस थिले तो स्ट्रक्चर काढी पारिबु, ठीक अछि। आ जदि भी तो पाखरे सीक्वेंस भी अवेलेबल नाही त तू लाइगेंड थ्रू रु तोर पूरा सीक्वेंस टा काढी कि तू स्ट्रक्चर बाहर कर पारिबु, बुझिल ना? तो
हाँ सही है ना मतलब
एवरीथिंग गोस
सीक्वेंस
कॉम्प्लीमेंटरी
सीक्वेंस प्रोवाइड
एग्जैक्टली।
जदि स्ट्रक्चर भी प्रोवाइडेड नाही ना सीक्वेंस अल्टीमेटली हमको प्रोवाइडेड अछि
तो स्ट्रक्चर करि पारिबु, मतलब बहुत
आराम से
सारा
सब
मेथड अछि देख,
स्ट्रक्चर करिया
होमोलॉजी मॉडलिंग अछि,
हम्म
अब इनिशियो मेथड अछि, तापरे तमरो एटा मतलब डॉकिंग भी अछि। तो
हम्म
मतलब वास्ट एटा कहूची ना, वास्ट एरियाज अछि जोटारे तू मतलब बसिकी घरे आराम रे कि
हम्म
तू मते स्ट्रक्चर दरकार तू एटा कर परिबु सेटा कर परिबु, बस टिके नॉलेज हबार अछि कि
हाँ
मतलब कोन रिमूव करी कि कोन ऐड करिबार अछि,
बस बस
आउ मतलब तते को फॉर्मेट रे सेव करिबार अछि, आ काहे कि सेटा सेमति सेव करा होची, सब
हम्म
रीजंस को देखि कि करिबार अछि, बस
मतलब
सेतिक।
एनालिसिस जदि करा जाउछि तारे कंप्लीटली करिया दरकार हाँ
हाँ हाँ हाँ,
सेट
कहीं ना अधा करले तोर रिजल्ट भूल आसीबो, आ जदि भी तू भाबिबो कि मतलब द रिजल्ट यू गॉट इज appropriate enough for a disease or for a drug designing that might be faulty in wet labs or performing experimentally, तो सेमती रे भूल आसपारे।
ओके
So anyone having any
Okay,
doubt?
anyone, if you want to ask anything.
Okay, so तमे ए पीपीटी थ्रू रू गोथरो तमे नीचे मतलब ऑटो डॉक करो तापरे यू विल गेट टू नो कि व्हाट हैपेंस नेक्स्ट मो पूरा प्रोसीजर गोटे पेपर रे लिखि देबी काहे ना ऑटो डॉक रे मु जति कि सेठी मेंशन करछि सेटा पूरा नाही मो पेपर रे मेंशन करके देबी कि मतलब केमती हाइड्रोजन बॉन्ड्स ऐड करबो वाटर मॉलिक्यूल्स काहे कि रिमूव करा जाए आउ तापरे पीडीबी क्यूटी फॉर्मेट रे काहे रखा जाए सब कुछ लिखि देबी तो सेमती रे तुम करो ट्राई करो देन शो मी द रिजल्ट्स कि केमती कौन आसला तमे को भी मतलब ये नहीं पारो किछि भी गोटे रैंडम प्रोटीन नहीं कि तमे करि पारो बट टिके मतलब एनालिसिस करि कि करले बेटर ओ ठीक अछि एनीवन एनी डाउट तो ग्रुप रे भी पचारि पारो यदि किछि डाउट रहला पीपीटी टा देखो देन विल जस्ट गो थ्रू
Okay, thank you, Samir, for giving this valuable insights.
अरे
এটা তো
मोस्ट
ଫ୍ୟୁଚରରେ ଆମକୁ ବହୁତ ବି କାମରେ ଆସିବାକୁ ଯାଉଛି। ଓକେ, ଯଦି ତମର କାହାର କ୍ୱେଶ୍ଚିନ୍ସ ଆସୁଛି ଦେନ୍ ପଚାରି ପାରିବ, ତାକୁ ଡାଇରେକ୍ଟ ବି ତମେ ପଚାରି ପାରିବ, କିଛି ପ୍ରୋବ୍ଲେମ୍ ନାହିଁ। ଓକେ ଓକେ
हाउ ठीक अछि देन विल एंड दिस सेशन अच्छा हाँ जदि काहाकू मतलब जीन थेरेपी रिलेटेड दरकार ताहाले ग्रुप रे थरे मेसेज कर देबो कपे माने सेटा आउट ऑफ डिस्क्रिप्शन अछि तो जदि जीन थेरेपी पर किछि दरकार मो पाकर पीपीटी अछि सेटा को थरो हमें डिस्कस करनबा आ जदि डाउट नाही देन वेल एंड गुड
ନାଇଁ କରିଦେବାଟା ବେଟର୍, ମୁଁ ଆଡ଼୍ କରିନଥିଲି ସେଇଟା ମୋର ହିଁ ଭୁଲ। ଜିନ୍ ଥେରାପିଟାକୁ ଆଡ଼୍ କରିଦେବା। ଓକେ
हाँ सेटा फाइनल रखिता
ହୁଁ
आ एंड कर दियो
ହୁଁ ହୁଁ ଓକେ।